This is a recorded lecture on blood composition and function, and lots of information about blood. If you've taken hematology, this should be a refresher for you. If you have not, I would recommend you take extra good notes and hang on to those for when that class comes around, because this is going to be kind of hematology topics in a nutshell, and then we'll expand on them significantly in that class. Blood. What is blood? Blood is a fluid, and it's a type of connective tissue. Kind of like cartilage is a connective tissue that's very specific, and bone is a connective tissue that's very specific. Blood is a very specific yet fluid connective tissue. There's different parts or kinds, I guess you could say. Whole blood. Whole blood is blood that's contained directly in the cardiovascular system. And peripheral blood, which is whole blood that's circulating in vessels, in blood vessels. Peripheral blood carries oxygen, nutrients, and waste materials. When you collect a blood sample from a patient, you are collecting a peripheral blood sample. And now plasma. So plasma is essentially the clear liquid in cellular components in which cellular components are suspended. In general, blood is red and opaque. It kind of has where you can't see it. It's not translucent or transparent. And it's made up of this plasma. And plasma, once again, is a clear liquid, primarily water. And then the cellular components are suspended. General functions of blood: transportation. They take oxygen to different tissues by way of hemoglobin, nutrients as well, transport different molecules of all kinds and enzymes. They take waste products away, carbon dioxide. They take hormones. Remember, hormones are from the endocrine system that travel from their producing organ to their target organ by way of blood. And then platelets, which we're going to discuss in detail. Regulation, body temperature. Remember that hypothalamus is kind of reading the temperature of the body, of the blood that's passing through it, and then it can instigate different techniques to either warm the body up or cool it down. Tissue fluid content, so it's able to regulate and know how much water and other fluids are in the body, in the system at all times. When there's not enough, it's considered hemoconcentrated, because the components, kind of the things of the blood, are more than the fluid is less. But if the fluid is more, that makes less room for the other components of the blood cells. That would be hemodiluted. Blood pH in normal is 7.35 to 7.45. You should know that. I would definitely write that down and know that. If it's not on your quiz, it's definitely going to come up in pretty much every single one of your other classes. So blood pH buffers acidic cellular waste products. We will discuss that further in respiratory. And then defense system. White blood cells, our own personal little army, and platelets to keep us protected so that we don't lose blood when we shouldn't. So cellular components of blood. So what that means is the cells that are floating around in that water plasma all the time in our body. So erythrocytes is the proper name for red blood cells. Leukocytes is the proper name for white blood cells, of which there are 5 types: neutrophils, eosinophils, basophils, lymphocytes, and monocytes. And also thrombocytes, which is the proper name for platelets. Now, the word hematopoiesis. Poiesis is the suffix meaning creating. Hemato is all blood. So, or all blood cells essentially, but blood. So this is creating all blood cells. How is it made? So hematopoiesis, the production of all blood cells. It's not specific to any one type of blood cell. And it's a continuous process throughout the animal's life. Cells have a lifespan, so they are constantly needing to be removed if they're getting old, aged, or defective, and replaced with new blood cells of a specific type. Now, as a fetus, the fetus produces hematopoiesis in the liver and spleen only. But after an animal is born and they're newborns to young animals, it occurs primarily in red bone marrow. Red bone marrow is also known as active bone marrow. In adults, as they start to grow, as you can see down here, the red bone marrow starts to turn into what's called yellow bone marrow. Basically, because there's less growth, replication, there's not as much need for as many cells, that red bone marrow starts to slow down. And then in adults, the only primary sites of red bone marrow are in the skull, ribs, sternum, vertebral column, pelvis, and the proximal end of femurs. The rest of what used to be red bone marrow in pretty much all of the bones is now yellow bone marrow, which is primarily made of fat. However, jot this down as well. Sometimes yellow bone marrow can reactivate in times of need. In some of those situations, not a sudden situation, but overall chronic infection and inflammation, things like that, the chronic anemia, etc., the yellow bone marrow is able to regenerate and reactivate into some red bone marrow to increase its production of hematopoiesis. There are a couple secondary sites for hematopoiesis also, and that is the liver and the spleen. It definitely can occur there, hematopoiesis, but definitely not to the extent of bone marrow, and it's really only in times of great need. Now, we're going to discuss pretty much this picture right here a ton in hematology. So what I want you to know of the basics is that all blood cells stem from this pluripotent stem cell right here. So this pluripotent stem cell is a very specific stem cell that can kind of turn into any cell that it wants. When it gets down to this next level, this hematopoietic multipotent stem cell, that's when it decides which direction it's going to take. So they all start as pluripotent stem cells. You do definitely need to know that. And then as it comes down the line, what I want you to get from this in this class is that red blood cells are one type. It can turn into red blood cells. There are different stages in between, but you don't need to know those. Or it can turn into a white blood cell, a granulocyte or an agranulocyte essentially. So neutrophils, monocytes, eosinophils, over here from mast basophil beginnings becomes a basophil. And then there's megakaryocytes, which shear off to make platelets, which I'm going to explain to you shortly. So from here, the different types of cells are made when a pluripotent stem cell turns into this type of cell it's going to be, and then matures into the cells that we know that are in peripheral blood. So starting with erythropoiesis, remember erythrocyte, that's your red blood cell. Poiesis means creating. So this is creating red blood cells, the process of creating red blood cells. There are multiple maturation steps like I discussed from that picture before, but you don't need to know those for this chart. Just know that they don't get into peripheral blood until they have matured most of the time in the bone marrow. The rate, however, of erythropoiesis is controlled by hormones. So what happens basically is as blood is flowing through the kidney and the blood recognizes that there is a slight anemia or not enough red blood cells in circulation, it lets out erythropoietin, EPO, from those kidney cells because with not enough, or hypoxia, I'm sorry. So if the kidney is recognizing that there's not enough oxygen or total red blood cells in the body, actually, it will release erythropoietin that will then travel through the body, because that's what hormones do, to the bone marrow where it will release more red blood cells. So this part down here actually means something to consider is when an animal has renal disease and it's not working properly, it won't be able to. If the kidney cells themselves are damaged and not working properly, they won't be able to release this erythropoietin in times of hypoxia. Therefore, we run the risk, and we often see anemia based just strictly on the lack of erythropoietin coming from the kidney. Something to consider. Leukopoiesis, so the creation of white blood cells, formation of white blood cells. That red bone marrow we discussed is responsible for production of all white blood cells. There are 3 types of white blood cell production, and that's just basically taking the 3 different main types of white blood cells and putting the word poiesis at the end. So there are granulocytes, so granulopoiesis, the formations of those 3 types of white blood cells, lymphocytes, so lymphopoiesis is the formation of lymphocytes, and monopoiesis, monocytes, and formation of those. The term leukemia means abnormal proliferation of one of the white blood cell types. It's considered a type of malignancy or cancer. What happens is when there's a cancer, it's usually a hyperactivity or a hyperplastic activity of the body of taking a certain type of cell and changing it to that it's malfunctioning it, and then it hyper replicates and makes a whole bunch more so that it kind of overtakes the good cells. That's kind of cancer 101 right there. Also, multiple myeloma. So that's caused by malignant plasma cells that accumulate in the bone marrow. So no good plasma cells, we'll talk about those at the end of this lecture. They start accumulating in the bone marrow, and then they crowd out healthy blood cells. So the things that are being kind of spit out of the bone marrow are unhealthy or defective, essentially. I wanted to note here that myelo is the prefix for bone marrow or spinal cord. So multiple myeloma this time is having to do with the bone marrow. Thrombopoiesis, so referring to the production of creation of thrombocytes or platelets. The stem cell that makes the cells basically turns into a stem cell that differentiates directly into a megakaryocyte. Megakaryocyte is kind of a bone marrow cell with multi-nuclei and things like that, and it's really big. What happens is that the megakaryocyte kind of pushes against the vessels in the bone marrow in these different parts, and the sheer pressure of the blood going by actually shears off fragments of the megakaryocyte cytoplasm. So platelets are pieces of megakaryocyte that are released into peripheral blood as platelets. Hold on one second, I kind of rearranged some of the order today. All right, so blood, primarily kind of talking about red blood cells, but all blood is a living tissue, even after you've taken it from an animal as a sample. So red blood cells in the body, they utilize glucose or plasma glucose for energy. So that plasma glucose that is floating around, basically blood sugar that we test when we, in some diabetic animals. But when it's been removed from the animal, glucose cannot be replenished in the tube. So the red blood cells still have to use it up for what they're doing. And then the glucose, there's no more glucose that it can pull from the plasma. Therefore, if you forget to spin a blood sample or you let it sit too long or something like that without spinning down, it can artificially decrease that blood glucose because the red blood cells are kind of eating it up and using it for energy. So it's really important to not let blood sit. Those set amount of times are definitely like that for a reason, and we need to make sure that we spin things down as they are supposed to be done. Functions of blood are definitely to, especially red blood cells, to transport oxygen into tissues by way of a carrier molecule called hemoglobin. So hemoglobin is basically the composition of a red blood cell that attaches to oxygen, and that way it stays inside the red blood cell and goes to the tissues to be used and extracted from those sites. Carbon dioxide also is then put into these red blood cells that are transported to the lungs to be removal as a waste product of the body systems of different cells. Red blood cells, remember, are called erythrocytes or can be limited to writing RBC. They're a highly specialized cell. The mature cell is what we see in peripheral blood, and it lacks a nucleus. It doesn't have mitochondria for that energy within the cell, which is why it gets its energy from plasma glucose or ribosomes. In appearance, it's non-nucleated. If you're seeing nucleated red blood cells, they are immature and called reticulocytes, which we'll discuss more of those in hematology class. They're called biconcave discs, meaning that they're circular and they kind of have two levels of circular area with a thinner central zone called a central pallor. These red blood cells, they stain red due to the hemoglobin concentration that's within the cells. If they stain really, really dark, they're concentrated with hemoglobin. If they stain really, really light, sometimes they can be iron deficient or hemoglobin deficient, which can be an overall problem for the patient. Red blood cells, or blood cells in general, being that they are living connective tissue, have a lifespan. Normal lifespan of each cell varies from species to species. Here's some examples of even just red blood cell lifespan. You don't need to know this for this class, but just you can see the smaller the animal, the shorter the lifespan. As it goes up to larger animals, larger or more days that it can live in peripheral circulation. You do need to know the term senescence, though. That's the process of aging. Cells can become senescent, meaning that they've aged and they lose their strength and their resilience in the body. Their enzyme activity starts to decrease. That cell membrane loses, it says deformability. That's kind of the ability to bend and move as it goes. Then it's at more risk of lysing or breaking down. About 1% of cells are removed from circulation every single day. They're constantly being made in these different processes that we've talked about, and they're constantly being removed, or this destruction happens by way of hemolysis. There's extravascular or intravascular, which is essentially blood cell lysing or bursting to clean them up and get rid of them and remove them from circulation. Okay, so this gets a little hairy for a second. So extravascular hemolysis and intravascular hemolysis, that's essentially the destruction of red blood cells, whether it's on purpose or not on purpose or immune mediated, etc. So extravascular hemolysis accounts for 90% of senescent red blood cells. That means red blood cells removed from circulation by macrophages. Remember those big eating cells that break things down and get rid of them? Clean everything up? Those macrophages are tissue macrophages from the spleen specifically. So this happens extravascularly, so not within the vessels in the spleen. So what happens when that goes on is these tissue macrophages phagocytize the red blood cells that are breaking down and are weak. When that happens, when a red blood cell lyses and breaks down, it's broken down into its basic part, which is primarily hemoglobin. Hemoglobin breaks down to iron and heme. Iron gets recycled, sent back to the liver, which will be used again to make more red blood cells as life goes on. Heme, however, attaches to different proteins like albumin, and it is taken to the liver to be conjugated and excreted. So when it breaks down into this bilirubin, it's unconjugated bilirubin, which kind of has this yellowish, brownish color to it. And when it goes to the liver, it conjugates it and sends it out of the body. Fun fact, this is the reason that urine and feces have their normal yellowish to brownish colors. And that process is very normal. If you go to page 300 in your book, there is a great paragraph there for you to read about extravascular hemolysis and that normal homeostasis of the body. Now, intravascular hemolysis, I'm going to have another slide on in just a second. So, but basically it's the other 10% of vascular hemolysis. So 10% of senescent red blood cells, old, aging, normal destruction. And that takes place within the red blood cells. So within the blood vessels, not red blood cells, my bad. So red blood cells membrane becomes weak and it ruptures within a vessel. I'll talk about it more in just a second, but the other kind of thing that can happen is you can get an immune-mediated hemolytic anemia. Immune-mediated meaning that it's mediated or provided or happening because of the immune system. The body's own immune system is kind of attacking red blood cells. and that's hemolytic anemia. So that ends up being anemic or have lower number of red blood cells in circulation because the immune system is causing this hemolysis. And this can be done intravascular or extravascular depending on the situation. So that's kind of the third option. 90% is normal by tissue macrophages in the spleen. 10% is normal, the destruction within the blood vessels by ruptured membranes. And occasionally things go a little haywire and they become immune mediated and cause an hemolytic anemia. A little bit more about intravascular hemolysis. I mentioned that it accounts for 10% of senescent red blood cell destruction. That destruction takes place within those blood vessels. So basically, the red blood cells are exposed, they're always being exposed to some type of oxidative stress. And that can be different pHs in the body, different things like that can disrupt the cell membrane and make it weak, misshapen, fragmented, things like that. Disease processes can do that. Some medications or drugs can do that. So there's many reasons to have oxidative damage. Now, when that membrane actually ruptures within the vessel, that hemoglobin, instead of being consumed and released in the spleen, is then released directly into the bloodstream. It has no major way to get all of it to the liver. So some of it binds with proteins in the plasma like albumin. It can get kind of to the liver. So a part of it gets to the liver, and then the process is kind of like the extravascular hemolysis at that point, how it's excreted. But any excess that is left in the bloodstream remains unconjugated hemoglobin. That would be called hemoglobinemia and is carried to the kidneys instead of the liver to be eliminated. It's eliminated through the urine, which would then be called hemoglobinuria. This can change the color of the plasma, and it can be change the color of the urine also from having this unconjugated hemoglobin or this broken down hemoglobin that causes this bilirubin in the urine in the blood as well. So platelets, proper term for platelets is thrombocytes. Thrombocytes are not complete cells. I kind of explained how those megakaryocytes are really giant multinucleated bone marrow cells, and they get pressed up into the holes of the bone marrow, and as the blood is surging past, it shears off these fragments of the cytoplasm and puts them into the peripheral circulation. So when you're seeing them on a blood smear, they are non-nucleated. They do sometimes have these, you can see in the picture here, some little dots from particles, but they don't have a true nucleus. They can be round to oval in shape, but don't get stuck on that because since they're fragments, they can really be any shape. They tend to be smaller than red blood cells, but they do vary by species. And macroplatelets or giant platelets can sometimes be seen too. It's not impossible. They have a clear cytoplasm since they are part of another cell's cytoplasm. And then you can see those blue and purple granules of the cytoplasm when it is stained. They also tend to be sticky, so you find them clumped. You should always check the feathered edge of your blood smear to look for platelet clumping if they've been activated at all, which we'll talk about the clotting factor cascade in just a second. And depending on your technique of blood smear, you'll hear all about that in hematology. Platelets have really important functions, most important for normal hemostasis. Normal hemostasis is basically preventing blood from leaking out. That's a matter of fact, that's what the definition is, AKA clotting. And it does that by formation of a platelet plug to be able to create a plug or the clot, but then stabilization of that plug. So it's not just like there's a clot formed and then that floats around in your bloodstream, because that actually would be dangerous. But getting that platelet plug to the site of injury and then stabilizing it in one location while things can heal. It's super fascinating. So first, in the clotting process, there is an injury. So basically the blood vessel is damaged, and the epithelium of the blood vessel or the lining of the blood vessel basically sends out a message saying that it's been wounded, and it attracts platelets to adhere to it. That's platelet adhesion. Then platelet aggregation and thrombin is kind of is kind of the same step a little bit, where platelets are connecting to make that platelet plug and stick together. So platelets get this enzyme. They're like, okay, this epithelium has been damaged. We need to unite. They kind of lock together and they head to the location that the enzyme is calling them to, essentially. In the meantime, or at the same time, fibrin, or the body, because of the presence of thrombin from this, thrombin is formed and it converts fibrinogen to strands of fibrin. So fibrinogen is a soluble plasma protein. So it's floating around in the plasma all the time. It's water soluble, but in the presence of thrombin, it turns into fibrin. And fibrin is an insoluble kind of substance, and it turns into strands. And that fibrin attaches to the platelet surface and essentially makes a cement. So that's that stabilization of the platelet plug. So platelet adhesion caused by the epithelium being injured of a vessel, skin, etc. That's what causes the formation of the platelet plug. That platelet aggregation, one more time, is those platelets that are stuck together. They stick together in one spot at the area of the injury. And then that thrombin being formed is converting that water-soluble fibrinogen to strands of insoluble hardened fibrin. And that fibrin is attaching to that platelet plug at the surface of the injury, kind of making a cement to lock it in place. So coagulation cascade is kind of that whole process. Technically, it's a series of reactions that result in 13 inactive enzymes. They are in there already in the body, inactive, and what happens is they start becoming activated by the preceding enzyme in the cascade. So the first one, or I guess number 13, depending on the way that you look at it, it gets activated by the injury, which then in turn activate the next, which then in turn activates the next, etc., so that it's like a waterfall. So they've all been activated, and that's 13 of those. On page 305 in your textbook, table 12.1 will show you those coagulation enzymes. You don't need to know their names or anything, but probably the quantity of those and understanding the concept. So the purpose of the coagulation cascade is to generate large quantities of that fibrin, that cement, to be able to create a patch to repair surfaces. So fibrin attaches to the aggregated platelet surface, so that big group of platelet that's in one area covering the injury, and that first of all plugs it and prevents further escape of blood, which we know that that's a big factor of clotting, is so that blood essentially stays inside the body or inside the vessels. Then as things proceed, this fibrin acts as a scaffolding to repair the damaged vessels. So the endothelium of the vessel produces the substances for fibrinolysis, which basically dissolves the clot when the endothelium is repaired. So this concept's like a scab. So you get a wound on the outside or the superficial part of your skin, and then you get a clot so that the blood stops coming out. That's your body's way of keeping the blood inside your body. And then it will harden and kind of dry up. That's that fibrin making that scaffolding and making that cement to keep everything together. And then as your body heals from within or by second intention, when it's done, you'll get kind of a dry, flaky scab that will itch and will essentially just kind of flake and fall off, and you'll have nice pink, new healed skin underneath. So that's the same thing that happens to our vessels inside to the body to keep them protected, so that way any poke or injury or bump or tear or anything like that isn't completely life-threatening. In the absence of platelets or impaired platelet function, that can result in bleeding disorders. So with now that you know exactly what platelets do, if there's not enough or they're not working correctly, there definitely can be some danger to the body. Usually the first sign of a platelet or a thrombocyte disorder is petechia. So that's small hemorrhages on the skin. Literally, it's making, if there's not enough thrombocytes keeping things in the body, red blood cells will kind of start falling out of vessels. Remember, vessels aren't like a PVC pipe. they're very permeable and they have these small holes. So fluids can start to kind of drip out or fall out if the blood pressure is too low, or red blood cells can kind of fall out and start pooling in places they're not supposed to if there's not enough thrombocytes in the body. Thrombocytes have a lifespan of about 5 to 7 days. The liver produces thrombopoietin to regulate the number of circulating platelets. So remember, the kidney produced erythropoietin that told the body to make more red blood cells. This is more specific to thrombocytes, the liver, and thrombopoietin. Macrophages in the body remove old or damaged platelets from circulation. And immune-mediated thrombocytopenia is when the body's own immune system is attacking the thrombocytes and getting rid of them, and then you have all these clotting type issues resulting from that. White blood cells. White blood cells, called leukocytes, or abbreviated as WBC. Mature white blood cells are what is in peripheral circulation, and they are generally larger than mature red blood cells when you're looking at them on a blood smear. There are 5 types normally present in that peripheral blood, and they can be broken down into 2 groups: granulocytes, which include basophils, neutrophils, and eosinophils, and agranulocytes, including lymphocytes and monocytes. Literally, granulocytes means a cell with granules, and a granulocyte means a cell without granules. So that's kind of the definition of those 2 words. An easy way to remember that is I say for granulocytes is grandpa Phil. So granulocytes, grandpa, and Phil. They have Phil at the end of their name. So basophil, neutrophil, eosinophil are all grandpas or granulocytes. That helps me remember that. White blood cells are kind of your personal army. They provide defense for the body against foreign invaders, microinvaders, parasitic worms, overgrowths of bacteria, viruses that have gotten into your body. They play a role in phagocytosis for getting and killing and destroying those foreign invaders, and they play a role in immunity as well. They are also produced in the bone marrow, just like red blood cells and thrombocytes. And they use peripheral blood to travel to their site of activity, which is why on a CBC, hold on, and each type has its own role, which is why when you do a CBC or complete blood count, it's important to know the total amount of white blood cells, but it's also important to count each individual type of white blood cell, because you might have a normal total white blood cell count, but individually you have a really high eosinophil count or a really high neutrophil count, which can be suggestive of certain disease processes since each type has its own job to do. So once again, white blood cells are kind of like your own personal army. The bone marrow is where it's produced, where they're made and they're trained and they're maturing, is kind of like the castle. And then the peripheral blood is the highway. So when they are sent out to a battle, they use the bloodstream as a highway, and then they're able to go into the tissues or sites of infection. It's pretty cool. So starting with neutrophils, one of the main granulocytes of white blood cells, they are 40 to 75% of circulating white blood cells total. They are the most abundant cell type for dogs, cats, and horses, which is kind of our primary thing that we discuss in veterinary, and then the other ones are a little bit more specialty. They have 2 to 5 nuclear segments, meaning the nucleus is segmented. You can see these pictures here. It's almost like twisted off like a balloon animal, and they can be different shapes. And 2 to 5 is considered normal. They're larger than red blood cells typically, but smaller than monocytes. Functions of neutrophils: they're involved in early stages of inflammation, the response to inflammation specifically. There's this term you should definitely know called diapedesis, and that's the process used by neutrophils to go from circulation into tissue spaces. So this is pretty cool. So you have a capillary and endothelial cells, and they have this ability to squeeze between the cells without lysing or breaking and get into tissue. So we don't want that for red blood cells because that's not their job or their function. We have them stay in with the thrombocytes to do their job, but certain white blood cells are able to get out of the circulation to the tissue. And that makes sense because if you have a skin wound, a liver abscess, or something very specific, we need the white blood cells to be able to go fight that off directly at the site. Another word, chemotaxis, is the process that attracts neutrophils to inflammatory chemicals at the site of infection. That basically means when there's an infection, when the endothelial tissue is infected or really damaged or sick, it kind of gives off an enzyme that brings neutrophils to it in this inflammatory process. Neutrophils' primary function is phagocytosis. So what happens is when they get to a microorganism, they basically engulf it and they create what's called a phagosome, which is kind of like a compartment, and they put the microorganism inside this phagosome. So it engulfs it, brings it inside, and then the cytoplasmic granules close off the edge, they move close to the edge of the phagosome, and then those granules have these different lysosomes, these different enzymes inside them that basically use like a chemical warfare inside the neutrophil to be able to kill this microorganism. It's like death by hydrogen peroxide. Basically, it has hydrogen peroxide type chemical inside, and then these lysosomes mess with the hydrogen peroxide and make it a deadly chemical to these microorganisms. And then it dissolves, kind of eats it up, then it moves on and does its own thing. So neutrophils fight by phagocytosis and cellular chemical warfare. Neutrophils only spend about 10 hours in the bloodstream before they enter the tissue. And the thing about them is that they don't return to the blood. So once they're made, they travel all around and then they get into certain tissues. So because they go into the tissues and don't return to the bloodstream, they are essentially kind of used up. All circulating neutrophils have to be replaced about 2.5 times every single day. And they're replaced by more mature neutrophils directly from the bone marrow. Kind of think of this army soldier as patrolling the tissues. This one goes through the tissues, hangs out where there's constant threat of microinvaders or where there's regular bacteria, in places like the lungs and the GI tract where there's always bacteria there and they got to keep it in check. Now, eosinophils are only 1 to 6% of total white blood cell count, so they're pretty rare in the peripheral blood smear. They're named for the red granules of mature cells when they're stained. So when you stain a blood smear and you find an eosinophil, you should be able to see the granules like you can see in this picture, and they do give off a pink to red hint or coloring. They do typically have a segmented nucleus, usually about 2 lobes, but really you can't go off of the lobes to decipher between a neutrophil and an eosinophil. They're typically slightly larger than a neutrophil, but that doesn't really help you if you don't have a neutrophil right next to it to compare. Also produced in the bone marrow, like all of these cells are, and they stay in the blood circulation for only 3 to 8 hours before they migrate to tissue where they remain. Changes in the number of circulating eosinophils, if there's an increase, that would be called an eosinophilia. If there is a decrease, that would be called eosinopenia, a decrease in eosinophils. Eosinophils have 3 very specific functions, or 3 key functions. One of those is inflammatory response. So basically, local, allergic, and anaphylactic response. Immunity, they play a role in immunity because they can ingest substances associated with immune responses. And phagocytosis, so similar to a neutrophil, but less on bacteria, but eosinophils especially are useful against protozoa and some parasitic worms. So for eosinophils, you'll see an eosinophilia sometimes in an animal that has a high worm burden or severe or chronic allergies. Basophils. So this is named for the blue granules of mature cells when viewed on a stained blood smear. In an appropriately stained blood smear, they should be really dark with blue circles, making the whole thing appear really dark. They are the least common white blood cell. Less than 1% should be in peripheral circulation. They are multi-lobed nuclei. They are similar to size in neutrophils. And the terms for changes in numbers of circulating basophils would technically be basophilia and basopenia. However, being that there should be less than 1% in the total white blood cell count, it's actually really difficult to quantify if something's normal or abnormal when it comes to basophils. Functions of this guy. Basophils are the least phagocytic of all of the granulocytes. What they have is in those big giant granules that they're full of, they contain histamine and heparin. So for histamine, they can initiate an allergic reaction. They can be like, oh, hey, there's something going on. We don't like it. We need to react. So it will actually initiate an allergic reaction, which then in turn should call over some of the eosinophils for anti-inflammatory processes. The contents of those granules of the basophil also contain heparin. If you remember, heparin is an anticoagulant, so it means the blood doesn't want to clot in that area. So that's kind of cool. So basically it releases heparin locally. So in that area that there's an injury or allergic, something going on, it will keep the blood from clotting and getting covered in thrombocytes and essentially allowing blood to continue to flow to the damaged area for repair. It shares some characteristics with the tissue mast cells. They both contain immunoglobulins, which we'll talk about more specifically in the immunity lecture. Lymphocytes. So lymphocytes are easily recognized because they have a really round or oval nucleus. Minimal, clear, almost colorless cytoplasm. It kind of tends to look like a big giant googly eye, in my opinion. It's pretty circular with a very large circular dark nucleus in the center. They mostly live in lymph tissue and circulate between the tissues and the blood. So this one goes from tissue to blood. The other ones tend to go to tissue and they stay there. Changes in number of circulating lymphocytes, the proper terms would be lymphocytosis for increase and lymphocytopenia for a decrease. Functions of lymphocytes. So more will be discussed in immunity, but kind of the basis I want you to know for this is that there are three major types, different types of lymphocytes with individual functions that help to regulate the immune system. So lymphocyte, lymphatic system, immune system are all kind of associated. Those three are T cells or T lymphocytes, B cells or B lymphocytes, and NK cells or natural killer cells. T cells or T lymphocytes, they're responsible for cell-mediated immunity. They are the most lymphocytes or the kind of lymphocytes that have the most representation in peripheral blood. So if you're seeing lymphocytes, pretty much they're going to be T cells, and that is responsible for cell-mediated immunity. B cells or B lymphocytes are rarely seen in peripheral blood. They're responsible for antibody production. Basics that you need to know for this class is that each B cell produces only one antibody type against one antigen. So basically, they can replicate a different type of antigen for immunity, and that's for the immune system is what's going to create either immunity or the ability to fight it. That's kind of what you need to know, is that it's responsible for antibody production, and each individual cell only produces one for a specific type of antigen. Lastly, natural killer cells or NK cells. They are granular lymphocytes that are found in blood and lymph, and they have the ability to identify and kill virus-infected cells. they actually bind to the cell and induce cellular changes that lead to cellular death. That's called apoptosis. So they don't phagocytize, so don't get that part confused. So basically they don't ingest the cell. That would release viral particles or different problems or tumor pieces because they fight off viruses and they work on tumors and cancers and things like that. But it's also called programmed cell death, which is pretty cool. So they literally attach and they literally change. It wouldn't technically be DNA that's inside there, but the makeup of the actual cell, they're able to change that and make it kind of a ticking time bomb so that it shuts down, kind of like a bomb. They're able to get into the center and cut the blue wire or whatever you see on the movies and makes it no longer work. So then it floats through, not causing the problem anymore. And then it goes through its regular lifespan and then dies and gets eaten by macrophages in the tissues and it's just gone. So not phagocytosis, apoptosis, and that is pretty cool about natural killer cells. Last but not least, don't worry about memory cells. Actually, what you need to know about memory cells is a clone of an original lymphocyte. And basically, when an immune response is needed, when a microorganism or something that has been exposed to for a first time, the body is able to react quicker and stronger. That's kind of the basis of immunity. But we'll definitely talk about that a lot more in the immunity lecture more specifically. Last but not least, monocytes. So monocytes make up of 5 to 6% of circulating total white blood cells, and they're the largest in size in white blood cell circulation. Nuclei can be a lot of different shapes. Don't get hung up on the shape of the nucleus. They can be round, they can be pseudolobed, so basically they can look like a neutrophil and they're not. They can look like a lymphocyte, but they would be way too big for that. So don't really stress about the nucleus when you're trying to identify them. They have an abundant cytoplasm, so they have a really large cytoplasm, and it stains gray. A lot of times, monocytes have vacuoles or kind of visual holes or what looks like bubbles inside them, and that's an easy way to help tell them apart. I literally call these in hematology sticky boogers, and when you get to hematology, you will understand why. Terminology for changes in numbers of circulating monocytes: increase would be monocytosis, and a decrease would be monocytopenia. Monocyte functions that you need to know are that it participates in an inflammatory response. So it actually goes pretty much straight into the tissues. It's obviously made still in the bone marrow like all the cells are, but then it goes into the tissues where it lives for over 100 days. Once it leaves the peripheral bloodstream, it's called a macrophage. It's no longer called a monocyte, it is now a macrophage. And a tissue macrophage, and then monocytes as they kind of converge and become the same thing, that is called MPS, mononuclear phagocyte system. They're basically the finishing crew. They're the crew that comes and cleans up the cellular debris after an infection or inflammation clears up. So let's go with infection. So there's an infection in a certain area. So we know that the other white blood cells go there. If there's a wound that thrombocytes have kept the blood in, kind of created that fibrin, scab, etc. So once that's all done, it's like, okay, well now it can't just stay there as like a bump on a log. So then these bigger monocytes come floating in and they get to that area and they are able to clean it up. So they're able to break down the debris. They're able to eat up any other pieces that aren't supposed to be there. And another cool thing that they do is they're able to basically send a message to the other cells, like, hey, you forgot some of these, if there's still kind of an infection brewing, and they can come back and finish the job. So the crew that comes through and cleans things up, make sure that the job gets done, the finishers are the monocytes and the macrophages as it enters the tissue. So it's pretty cool. All right, that is a lot of information about blood. I hope you learned something awesome.